Hi everyone. We're in section 7.1 and we're in microbial metabolism. And just to give you a quick definition of metabolism, we all talk about our metabolism. I've got a fast one, I've got a slow one. But really what metabolism means is it's all the chemical reactions of the cell. So here it says there's thousands of different reactions and they fall into generally one of two categories. We either call them anabolism or catabolism. So metabolism are all the chemical reactions in a cell and ultimately all the chemical reactions in an organism. And they can be described as some of them are part of anabolism and some are part of catabolism. Turns out chemical reactions that are called catabolic reactions are a part of catabolism take large molecules, break them down into smaller molecules, and release energy. You can think about the chemical reactions that are related to digestion, to us acquiring food. Why do we eat food? Because we need energy. So we take large molecules, our digestive system breaks them down into smaller molecules, and we release energy. Anabolism are the chemical reactions that take the smaller molecules. A lot of these were made in catabolism, and assembling them into larger molecules. And this requires energy. So you can imagine that catabolism and anabolism work together. Catabolism, large molecules break them into small molecules, releasing energy. We're using those small molecules to build into bigger molecules, and we're using that energy to help build it, and that gives us anabolism. And that will allow cells to grow and to repair parts. I don't think you'll know the answer to this question until we get a little bit further, but you should be able to get that. Here it just gives a little summary of metabolism. What does metabolism do? It assembles smaller molecules into larger molecules. So it's small into big, that's anabolism. In this process, ATP is utilized. So when I say energy and metabolism, we're usually talking about ATP. And I think most people are familiar with that term, that ATP is a molecule. It's an energy carrying molecule. It's important for chemical reactions in cells. So it says anabolism. And then metabolism also degrades the large molecules into small molecules, releases energy, that's catabolism. And then that energy is transferred in the form of ATP. This diagram is just trying to show you, if you look here, we're going to spend the most time on catabolism, this yellow part. But right here we have catabolism, and this is complexity of molecules. So we start off with larger molecules, and through catabolism we're going to break them down, and it's going to yield energy. It's going to yield ATP. Now we have some of the building blocks, the molecules that are used to make things like glucose. And anabolism is going to use those, and it's going to assemble them into larger and larger molecules and ultimately into entire organisms. So anabolism is building up, catabolism is breaking down. I wanted to talk a little bit about enzymes, and for a lot of you this might be review, but it's just a good refresher on what are enzymes. Remember we said enzymes are primarily proteins. It says here are enzymes are made of protein or RNA. We're not going to talk about the specialized RNA enzymes, but we're going to talk about the ones that are made of proteins. So most enzymes are made of proteins. Chemical reactions cannot occur without enzymes. So if we're talking about metabolisms, all the chemical reactions in a cell, you obviously have to have lots of enzymes. Enzymes are what we call catalysts. Catalysts speed up the rate of a chemical reaction without being used up itself. So, and without these enzymes, we would not be able to conduct life. Here's just general characteristics about enzymes. I'm going to say most of them are made of protein. They speed up chemical reactions. They have unique characteristics, such as shape, specificity, and function. There's lots of different enzymes that do lots of different things. They can speed up chemical reactions to be compatible with life. We're going to talk about the active site and substrate and how they're not changed. they can be used over and over again. So you don't need a lot of an enzyme to do its function. They're affected by temperature and pH, and that's because they're proteins. Proteins change their structure when the temperature and pH change. And we're not going to really talk about regulation, but that's true. So I wanted to give you like how do enzymes work? And he has some vocabulary here, and I think I'm best if I just show you the picture. I'm going to go down here. Here we have, in fact, let me enlarge that a little bit. Here we have an enzyme. So an enzyme's a protein. This is just a generic enzyme. And so here we have an enzyme. And one of the things, remember, enzymes are proteins. Proteins have very specific shapes. So here's two molecules. And the molecule that the enzyme works on is called its substrate. And enzymes can be very specific to their substrate. That's why we need lots of different enzymes doing lots of different things. And it's showing you these two molecules here. This one fits into the enzyme, but this one doesn't. And the space where the substrate's going to fit kind of matches the substrate in some way. And we call that the active site of the enzyme. So here's our enzyme. Here's the molecule the enzyme's going to work on. It's its substrate. This is just some kind of molecule that doesn't fit in there, so we're going to ignore that one. When the enzyme and its substrate come together, we call it the enzyme-substrate complex. The chemical reaction occurs, and the enzyme somehow changes that substrate. It might put something together, it might break something apart, or just change the shape of the substrate itself. But here's the products. And now you look at that the substrate changed into products, but the enzyme itself stayed the same. So that same enzyme can go interact with another substrate. So this thing can go around and round. I'm going to jump to another diagram that's not in the book. I'm going to go to the learning outcome number 4, explain what happens to an enzyme when it's been denatured. So in the previous diagram, we were looking at enzymes. We said enzymes are proteins and they have distinct shapes because they need to have an active site and the substrate needs to fit there. So if we have an enzyme, in this particular case, here's the native protein. This is the natural enzyme. And let's say the active site is this area right here and that a substrate fits in there. And so the shape of that enzyme is going to be really important. If our enzyme is what we call denatured, which means unfolded, you will see that you still have your amino acid chain. Remember, proteins are chains of amino acids. You'll still have that chain, but it's not going to be folded in its normal way. So if you no longer have this active site, you're no longer going to be able to interact with that substrate and the enzyme will not function. The kinds of things that can denature proteins are all sorts of environmental conditions, including extremes in pHs, changes. I wanted to mention temperature because we not only use temperature to affect enzyme activity, but we use it to kill bacteria, and it actually has to do with proteins and enzyme structure. I'm going to skip to a diagram that I found, and this isn't in your book, but it's something that you can find easily. Oh, I have it in your lecture study guide, actually. So here we have enzyme activity. Here we have temperature, increasing temperature. A is where the temperature at which the enzyme works its best. And an enzyme that's in bacteria that's associated with the human body will probably have an optimal temperature of about 37 degrees Celsius, the human body temperature. When we think about denaturing proteins, and I think about egg whites. When you look at an egg, it's a raw egg. The white part's called the albumin, and it's kind of clear and jelly-like when it's refrigerated. Once you cook it, it turns opaque white. And so that's a really good example of what a change in proteins when they denature. So in colder temperatures, the albumin is very globular and you can see light through it. And when you denature it, you unfold those proteins and they all kind of line up and they become opaque. So here if I have low temperatures, you notice we have low enzyme activity. Well, we know that from egg whites that the protein is not denatured, but we have low activity because under colder temperatures, chemical reactions are slower. Here we have at our optimal temperature, the enzyme's working the best. And now we get above that optimal temperature, and now it gets too hot for the enzyme and it denatures, it unfolds, and therefore we don't have any activity at all above the optimal temperature. So these low temperatures are going to just slow down the chemical reaction of that enzymatic reaction. High temperatures are actually going to denature the protein in the enzymes and completely prevent it from functioning. Sometimes I'll put a question on tests and it will say, which one of these is not an enzyme? Multiple choice. And I list all these crazy words, and some students will say to me, wait a minute, I didn't know we had to know all these words. And it's kind of a trick because when you look at the names of enzymes, so I have an objective that says, explain how enzymes are named. When I look at the names of enzymes, somebody had a great idea and said, let's take enzymes and end their names in ACE. So there's a lot of science words that end in ACE: carbohydrase, lipase, proteinase. And generally, the enzyme name is going to tell you what it does, and it's going to tell you it's an enzyme by putting an ACE at the end. So a carbohydrase probably breaks down carbohydrates. A proteinase or a protease breaks down proteins. Maltase breaks down maltose. And here we have a polymerase. It makes polymers, makes chains of things. So it's kind of handy. But there was one smart aleck guy that said, well, wait a minute, I've got a couple enzymes that end in IN. So every once in a while you hear of an enzyme that ends in IN. The one I can think of that I hear the most of is renin. And it's an enzyme that causes milk to clot in the calf's stomach, the stomach of a calf. And renin has been used and continues to be used to this day to help make cheese in some cases, helps the milk clot a little bit better. But naming enzymes, look for that ACE or even look for an IN. The last thing I want to mention in this section is the concept of a metabolic pathway, and we're going to talk about some metabolic pathways, but I just want to mention, like, what the heck does that mean? In a metabolic pathway, let me just back up and say enzymes don't function alone. They generally function in conjunction with other enzymes. Think of an assembly line. We're going to talk about how we take our food and extract energy, and that happens in many, many steps. It doesn't just happen in one big enzyme does everything. So this is illustrating what some metabolic pathways look like. So in a metabolic pathway, the product of one pathway, the product of one enzymatic reaction is the substrate for the next. So here I have substrate A is here, the arrow represents an enzyme. There's an enzyme, and there's its product, B. Now, B now becomes the substrate for the next enzyme, and there's that enzyme's product. C now becomes the substrate for the next enzyme, and onward. And you can see there's all different patterns of metabolic pathways. But when we're talking about a metabolic pathway, we're talking about a series of enzymatic reactions where the product of one reaction becomes the substrate for the next.